And we started out with this premise that xenobiotics and their metabolites comprise a significant fraction of the non-confirmed or uncharacterized low abundance signals in high resolution mass spec analysis of human samples. Given that premise, immediately at least two problems arise. One is that because metabolomic data provide only snapshots in time, their exposures of xenobiotics are often defined not by the parent compounds, but by their metabolites, as the parent chemicals are rapidly eliminated by liver enzymes. So this on the right here is just a pharmacokinetics of bupropion in mice dosed with bupropion. And what you can clearly see is immediately after the injection, you've got high levels of the parent compound that rapidly declines, so that by out towards 8 hours, there's very little of it left, whereas the major metabolite hydroxybupropion is generated from that and persists much longer. So this is going to be the main signature in a snapshot out at this time. Secondly, these metabolites are often, or even usually, very low in abundance, too low in abundance for high quality mass spec, MS2 spectra to be obtained. And also coupled with that, the authentic standards for these metabolites are not available. Some of these metabolites may not even have been characterized before. So these metabolites can't be identified by mass spectrometry criteria alone. And so what we basically hypothesized is that authentic xenobiotic metabolites produced in the laboratory from human liver enzymes can be used as novel biological criteria, or surrogate standards, if you will, to increase confidence in compound ID. And a secondary hypothesis is that co-occurrence or correlation of these xenobiotics with their metabolites in human samples can be used as an additional criterion. So our strategy was to use our expertise and knowledge of xenobiotic metabolizing enzymes to develop tools to generate authentic metabolites at scale for use in identification. And we wanted to concentrate on MS1 spectra, again, due to the fact that there are low abundances of these metabolites in human samples, and MS2 spectra are not going to be practicable in many cases. So we started out using pooled human liver S9 supernatants that comprise the endoplasmic reticulum and the cytosol of the cell. These contain all the important phase 1 liver enzymes like P450 enzymes and phase 2 enzymes like UGT glucuronidase transferases. And so we developed a platform using these liver S9 supernatants that I'll talk about, and also developed a panel of hepatoma cell lines, human hepatoma cell lines, expressing individual human xenobiotic metabolizing enzymes. And these can be used for production of metabolites that you couldn't detect with S9 supernatants, and to scale up reactions for producing more metabolite for more conclusive ID. We did this in 96 well plates because, as I said, we wanted to do this at scale. And the idea was to generate tools to in turn generate a database of authentic metabolites from thousands of xenobiotics with retention time and MS1 spectra as primary values, but also MS2 spectra where available. And then go on to use these metabolites to identify them and their parent compounds in human samples with documented and undocumented exposures. So this is just a sort of diagram of that overall concept. Here we've got our S9 supernatants with various chemicals incubated in 96 well plates for different times. And we're going to subject those incubations to LCA HRMS analysis. Those are going to detect features that are increased, that emerge or are increased over time, so are probably metabolites, and that's coupled with bioinformatics analysis, prediction of likely metabolites by biotransformer and other bioinformatic methods. And so we generate this library of laboratory generated bona fide metabolites. And then using these profiles or fingerprints, we can go in to human samples analyzed in the same platform and detect them by their retention times and M to Z values. So how did that work? Well, to start with, we need to show that the platform works. So this is generation of expected metabolites from well-characterized compounds using the S9 system. And in the left here are results for one such compound, beta-naphthoflavone, where you can see two hydroxy phase 1 metabolites generated over time, and also two phase 2 conjugated metabolites, the sulfate and the glucuronide, also generated over time. And then the heat map on the right kind of summarizes the data for all the compounds that used in this small preliminary study, where we found that we were able to detect about 90 to 95% of all the major expected metabolites from these prototypic compounds. So then using that kind of data, we did what I said we were going to do, which is to go into in vivo and see if we can detect these metabolites in both mice and humans. And so up here in panels A, B, and C are essentially the data. The panel A is essentially the data I showed you before for bupropion in mice injected with bupropion in vivo. You can see the rise and decline of the parent compound, and the black are the different time points in the mice, and the red is the in vitro S9 generated authentic metabolite. And you can see that we indeed detect this metabolite 2,3-hydroxybupropion, as well as hydrobupropion in these animals. And then going into humans with documented bupropion exposure, we accessed a cohort of people participating in a clinical trial at Emory whose drug exposure was documented. And we tested all of them for bupropion and its metabolites. And what you can clearly see is that, yes, the parent compound and both metabolites were identified in all three samples from this individual, and only in this individual who was the only one taking bupropion. And the networks over here on the left describe the same kind of data for other drugs that people in this cohort were taking: acetaminophen, metoprolol, carvedilol, and warfarin. So what's a little bit more of a challenge is to do the same thing in humans with undocumented exposures. And so we took a panel of metabolites that we had generated to that point. I think we had about 30 odd at that time. These are results from 4 of those selected xenobiotics. So if we start here, this is so going across here, 120 different individuals, adults, participating in the Emory Georgia Tech Centers for Health Discovery and Wellbeing study. And what you can see is, if we look in the paired human urine and plasma samples, that there are 1, 2, 3, 4, 5, 6, 7 individuals, right, who consistently display nicotine and all of its metabolites, cotinine, the glucuronide, and the hydroxycotinine. So this is very strong evidence that these individuals have been exposed to nicotine, either by smoking or through environmental exposure. And this diagram also illustrates an important point of this approach, which is that we have apparent levels of cotinine in the vast majority of these individuals, right? Some of them without accompanying nicotine metabolites. So if you were just looking at one metabolite, you might think that all these people were exposed to nicotine, but in fact, cotinine has the same molecular mass as serotonin, and in our system runs right around the same retention time. So it's really important to have these corroborating compounds, right, all in a network together to document that exposure. Below that, we have naphthalene, and we detected naphthalene sulfate, hydroxynaphthalene sulfate, and hydroxynaphthalene glucuronide, right, in paired samples from 3 individuals in this cohort. Again, very good evidence that these individuals have been exposed to naphthalene. This also sort of illustrates the point I made earlier, which is here's a compound for which its exposure is not really well detected if you're looking for the parent compound, but for these 2 phase 2 metabolites, you can detect them using those. I won't spend any time on the omeprazole data, but just going down to the bottom here for piperine. Now, piperine is a component of black pepper. And if you look at the piperine signal, actually, we were able to detect some kind of signal for piperine in all 120 participants in this study. But for bona fide or let's say higher confidence of piperine exposure, we also look for the metabolites of piperine dihydroxy and the piperine dihydrodiol. We can identify several people who obviously have a higher level of exposure to this than others. But again, important to have the corroborating evidence of piperine metabolites, because piperine has the same molecular mass as morphine. So now I'm going to spend a little bit of time telling you about our second platform that we've been developing, which are hepatoma cell lines expressing human xenobiotic metabolizing enzymes. And in the latter stages of the project, we focused on cytochrome P450 enzymes because those were the ones that seemed to be of highest activity in these cell lines. And this work I'm going to describe was published is published in Drug Metabolism and Disposition. We just got informed this morning that the manuscript was accepted, so that was nice. We're looking here at a Western blot of expression of CYP1A2, 2A6, 2B6, 2C8, 2C19, 2D6, 2E1, and 3A4. So they're all expressed robustly in these cells, shown by the red bands. And down below here is a demonstration that each of those enzymes is catalytically active with well-defined substrates of these enzymes, and indeed that they exhibit the expected substrate specificity. So for instance, for chlorzoxazone, you only see activity with CYP2E1. Moving on, we wanted to quantitate, how active are these enzymes? So we wanted to quantitate it. We got the authentic standards for hydroxybupropion, its major metabolite, and quantitated production here over 24 hours in these incubations. With 100 micromolar bupropion incubation, we were able to retrieve 50 micromolar hydroxybupropion. So this is an illustration that these are very powerful tools to generate lots of P450 derived metabolites. We were also interested to detect another metabolite of bupropion called dihydrobupropion at very low levels, but nonetheless consistent in these cells that we could not detect in the S9 incubations, presumably because of the low activity and lower expression of the enzyme. And we were, this is a bona fide in vivo metabolite. It had been reported only once before, I believe, that we could find in human urine samples, but the source of that metabolite was not known. But now we can say that it's generated by CYP2B6 in the liver. And going back into those mice injected with bupropion, we were able to detect the time-dependent generation of the dihydro metabolite and its subsequent elimination. And going into that small cohort of humans, of whom one was exposed to bupropion, we were able to detect it in that individual. And here is the MS2 spectrum from that signal in human serum in blue and the CYP2B6 cell line in red. And so, we want to think about using these cell lines to also generate metabolites at some degree of scale. But when you think about, basically making metabolites from 36 chemicals from 8 different cytochrome P450 expressing cell lines with 2 different time points in duplicate, that becomes a lot of samples and a lot of mass spec time. And so, excuse me, I'm just checking my time here. We decided upon a pooling strategy to basically increase throughput and get around that mass spectrometry bottleneck in this study. So what we did was we divided these 36 chemicals into 6 pools of 6 and pooled them as shown here in the left. And then we basically flipped that matrix so that the columns became rows. And so we had another matrix here. And so every compound is present in 2 different pools, but in each pool, the compounds it's there with are different, right? So this was a good sort of internal way to verify the specificity and generation of the metabolites. On the right here are just some extracted traces from a couple of the prototypic compounds that we again included in this study. So you see hydroxy beta-naphthaflavone from CYP1A2 and hydroxytolbutamide from CYP2C19. And you see these are only 2-hour incubations with the cells. And interestingly, something I'm going to focus on in the next slide is the metabolite dihydroxytrifluoralin from the pesticide trifluoralin, which is a pretty ubiquitous pesticide whose human metabolism hasn't really been characterized till now. And so shown here in this heat map are basically details for the metabolites we detected for 4 of the 36 compounds. And I wanted to focus initially on trifluoralin. So what you can see here is generation of trifluoralin in basically metabolites in basically 4 of these wells, right? So these are wells containing pools, metabolite pools that contain trifluoralin. We get no signal from pools that had no trifluoralin. Also, the metabolism was P450 enzyme specific, so these metabolites were formed by CYP2B6 and to a slightly lesser extent CYP2E1. So we're showing enzyme specificity, but also generation of these metabolites. And we got this predicted metabolite of dihydroxytrifluoralin, but we also saw 2 features that we're pretty sure, whose MS2 spectra I'm not going to show you, but are consistent with actual metabolites of trifluoralin, but we don't know what they are. And so we're currently actually scaling up, or we're working on scaling up CYP2B6 cell incubations with trifluoralin to generate enough compound of these compounds for more conclusive or some sort of chemical identification. Same story with pazopanib here. The metabolites were detected only in wells that had pools that contained pazopanib, and it was the generation of these 2 metabolites was specific to CYP3A4 and 2C8. So I won't go through the last 2, but you get the idea. So to summarize this part, and we're almost finished, through enzymatic generation of xenobiotic metabolites by 2 different enzyme systems, we've generated platforms that can generate metabolites from thousands of chemicals. And in fact, we now have actually a metabolite library from more than 1,000 xenobiotics. And these, the mass spectra from these, mass spectral data, are retention time in MS1 and MS2, where we can detect it, are basically being translated into an organized computational framework that's suitable for community databases like Metabolomics.cloud that Shizao has generated, Mona and MASST. We've also provided evidence that these enzyme-generated xenobiotic metabolites can be used as surrogate standards to identify them in human samples with matched retention times and MZ values. And together with co-occurrence or correlation of parent chemicals and their metabolites, they provide practical criteria for biology-based compound ID. And by combining the power of these xenobiotic transformations with computational innovations, we think this can form a basis for developing strategies for high confidence personalized exposomics. I'll quickly go over our future directions. We're finishing basically screening, we call it screening, but generating metabolites from a spectrum library of 2,560 different xenobiotics for metabolite production. Shizao's group is going to continue developing xenobiotic metabolite relevant databases and build spectral libraries for these environmental chemical metabolites, testing the functionality in cloud environment using Docker containers. And there's a prototype expected of this towards the end of the year. Grant Singer, tomorrow from the group, is going to present a lightning talk on a unified workflow for a biology-based exposomics. So, and we hope to have we plan to have a manuscript submitted on that by the end of this month. As I said before, we're working on scaling up trifluorine metabolite production for purification of them and the chemical identification. Working on expanding our analysis to include MS2 where available, refining and optimizing the pooling strategies to increase out throughput of metabolite ID even further. You know, is 6 the limit? Is 12 the limit? Is 24? We don't know. And we also plan to work on metabolism of complex mixtures such as natural products and air pollutants to characterize their exposures in people. And finally, Shizao talked a little bit about this yesterday. He's developing his lab is developing a fast algorithm to search xenobiotic signatures in databases called XASARI, which is based on the ASARI program he developed and described yesterday. And they're using HBM4 EU CE screen database of chemicals of emerging concern from the EPA and their metabolites, and using our in vitro generated metabolite data to validate that computational method. And this work's still ongoing, but Shizao finds that they can using XASARI, they can search a full database of 140 compounds in about 30 seconds, which makes this a very feasible tool for large-scale exposome data mining. And that's it.
Thank you, Eddie. That was very nice. We do have about 4 minutes for questions, and I can get us started with one. Looking at the kind of human samples, you said, using the not just cotinine but the other metabolites lets you know who is actually exposed to nicotine. How much of that is dependent upon the kinetics of the metabolism versus dosing, and the sensitivity of the detection? So it becomes very complicated. And how much are you using kinetics as part of the analysis there?
Yeah, I mean, I don't think we can use kinetics unless we have serial time samples, right? In the time frame in which nicotine is metabolized, and it's a very good point. That kinetics, time dependence is an important consideration in analysis of any metabolomic data. At this point, we haven't really taken it to a quantitative or even semi-quantitative, level. at this point. I think that's, you know, something we can think about going forward. But at the moment, we're really just looking for, you know, co-detection essentially, or correlation, without worrying too much about, you know, the relative levels of each metabolite, which might be very different, right? So nicotine is metabolized by CYP2D6 and 2A6, sorry. And there's a genetic polymorphism in there where there's people are very fast metabolizers, people are very slow metabolizers. So the signature is going to be different in different people with different genetics. So that would be a really interesting use of the tool, actually, would be to look at, to use it to look at genetics a little bit.
So David, I could also comment on that. When we were looking at secondhand exposures, where there were individuals with documented secondhand exposures, we didn't pick up very many. We pick up cotinine and, you know, sometimes norcotinine, sometimes hydroxycotinine, but at those lower levels, it really wasn't nearly as reliable as at the high levels.
Yeah. That's kind of what I figured. You know, it may be a discriminator between primary and secondhand smoke as well.
Yeah, so I mean, you know, I think, you know, the more metabolites you have, the better, right? Because that kind of mitigates the kinetics to some extent. And that, you know, okay, if the parent compound is gone, you might still have 3 metabolites that corroborate each other in terms of the exposure.
Tom?
Yeah, great talk, Eddie. And I'm a fan of the approach that you guys have developed and are using there with the in vivo metabolism screening. What percentage of the, you know, exposome do you think you've covered so far? And is the plan to just keep going, you know, through all of the exposome molecules? I don't know how easy it is to get authentic standards, you know, for incubations for all of these. And, you know, if I ruled the world, I would say that any chemical manufacturer would have to set aside a pure substance and offer that to the scientific community for monitoring, as well as an isotopically labeled version of that. But, you know, just thought curious how far are you guys going to take it?
Well, you know, the grant is ending, right? And so we need to find funding to take it further. And since I'm actually closing my lab at the end of the grant, I'll leave it to Dean to answer that.
Okay.
So the plan is, I mean, we've been working with EPA to get the, you know, their collection, and we've analyzed them all, but Doug Walker is going to be coming back to Emory, and we have discussed the plans for that to use the high throughput to try to generate. What we're doing right now is combining the GC analysis, where we get a lot of the environmental chemicals, with the LC data, looking for the correlation of the metabolites. It certainly works for you know, some of the polycyclic aromatics and so forth, but we haven't gotten very far on that. But definitely it's in the plans, and hopefully others will do this because it's extremely powerful.
Yeah, I like it.
And Tom, to your point, that was another point that was made at the CTAC meeting last week. Particularly the Europeans were pushing very hard for the chemical industry to make authenticated standards available for research purposes. I agree. I wish we could do something about that here.
Yeah.
So on that note, we'll switch over